Okay, so welcome to lecture 22. Just a couple of important notes. So for Friday's lecture, normally Kim Curry comes around and talks to each of the sections, but she's unable to do so this year. So she has posted her lecture about writing your final report, your scientific report. The lecture is, I think, about 50 minutes. So instead of coming in and listening to it, she's posted it onto the Brightspace site. So you're expected to watch that lecture, and you'll need that information for writing, or at least for doing a good job on your report. So instead of having it as additional material to watch, the two of us decided we would just have it delivered online. So there won't be anyone in class on Friday. Well, I mean, you're welcome to come in, but importantly, I won't be here on Friday. I'll put feedback up probably tomorrow for the midterms, and then starting next week during office hours, I'm happy to go through them and discuss any concerns or anything you have about them. So again, the class did a lot better this time, and hopefully for the final we do even better again. So come and talk to me if you have any concerns whatsoever. If you can't come for office hours, again, I can meet with you outside of office hours. It just sometimes a bit of back and forth figuring out a time that works. The third thing is totally weird, and it's going to upset our lecture today because we had a fire alarm in the morning lecture. So, and as I said, I want to make sure the two sections are covering the same material. So I'll make some loud fire alarm noises at about, it was about 5 or 10 after or something. So we lost about 10 minutes of lecture. So you can have an extra long coffee break between your classes. So sorry about that. I don't know, it seems like there's a lot of fire alarms at UVic this year. So I've taught here for 13 years, and I've never once had a fire alarm ever in a lecture, and I've had 2 this year already, so yeah.
I thought they were just testing the fire alarm system.
They didn't give us any warning of it. So I don't understand why it's worked out that way this year. So normally they give us heads up if there's anything scheduled, but I think they are tests, but I'm not positive. So anyway, it's a mystery. Okay, so let's get to it then. All right, so we were looking at endo and exocytosis in the last lecture. And endocytosis, 2 things can happen here. We can bring things in non-specifically. So components that are just kind of hanging around the surface, the plasma membrane of the cell, can be pulled in. Okay? And so anything that's sort of associated with where that coat starts to form, if that's maybe going to form, well, here, I guess would be, those can be brought in. And that's penocytosis. We can also have receptor mediated, and there's around, I believe, 25 receptors which have a specific ligand. When we talk about a ligand, all that is something that binds to something else, okay? And so the example we looked at was bad cholesterol, LDL as a ligand. Once something enters via an endosome, there's sort of 3 pathways it can take. Often what we associate them with is going through the endolysosomal system to the lysosomes. And in the lysosomes, those products that are brought in can be degraded, broken down into individual components, or sometimes other modifications occur. And one of the examples I talked about was how cholesterol is actually in a form that's not usable until it goes to the lysosome, and then it's processed. They can also go backwards, which isn't shown here, through the Golgi, even to the ER and the cytosol. So they can use the retrograde transport as well. And finally, they can be used for transcytosis. And in transcytosis, it's a way for components to journey through the cell. And so we would see this on cells that are called polar cells, where they have 2 different sides, basically. So the example we looked at was an enterocyte, which has an apical region that it projects into the gut lumen, and a basal, which is inside the actual body. And it can move things from the gut through that barrier the gut barrier. That was mediated by clathrin. We talked about other coproteins, however. And the thing about coproteins is that they will be interacting at specific membranes and used for certain purposes. And they'll associate with their own complement of receptors. And so clathrin, where we see it then, is forming on the plasma membrane, but also going out from the Golgi. In contrast, COP2 goes out from the ER towards the Golgi, and COP1 is involved in retrograde transport. And the way these coproteins work is that they will spontaneously and favorably form these structures. And if you want to do something unfavorable, like bleb off the membrane, you have to join that or coordinate that with a favorable activity. And so here, these coproteins will naturally form structures. This one is clathrin, and it's favorable. But remember, these coproteins are associated either directly or indirectly with the membrane. So as they form, they pull the membrane in. And we can see an example of that here. The thing to keep in mind is where these coproteins associate with the membrane is regulated. And it's mediated by GEFs for either ARF or SAR1, for the 3 examples we looked at. And those GEFs cause exchange of monomeric GTPase that is the site of initiation of coat formation. There's a further input of energy by dynamin, and what dynamin does is that squeezes the vesicle, pops it basically off the parent membrane. And so that will cause the parent membrane the membrane to seal up on the vesicle, and also the parent membrane from which the vesicle arose. And so that does require an input of energy as well, because we're moving polar head groups through a hydrophobic space. And so again, where this occurs is mediated by these different GEFs, either ARF-GEF or SAR1-GEF. And finally, we talked about fusing membranes, not butting them off, but fusing them, which again requires an input of energy. And in this example, that energy comes from snares. And snares will form these 4 helical bundles, which are very stable and very energetically favorable to form. And really what literally happens is there are snares on the surface of the vesicle and the target membrane. The targeting is mediated by RABs, which is a large family of monomeric GTPases. I think around, can't remember what we said, something like 70 members. And it's also mediated by these tethering complexes. And when those snares come into close contact, they form that bundle. And what that does is it actually just pulls the vesicle tighter and tighter and tighter to the target membrane until it causes fusion. So a really mechanical process. And I actually found a good video for this. It's quite short, and I posted that on Brightspace. So you can have a look at that if you. It's showing just a model of how the fusion occurs and how it's mediated by the snares. So we're going to look at our ion permeability, 2 different equations, the Nernst and the Goldman equation, and then steps in synaptic signaling. And we will actually get as far as that today, and then we'll have a few things to pick up in our next class. So again, we're looking mostly here at neurons. And so the neurons in the glial cells are the 2 main components of the peripheral and central nervous system. The neurons are sort of the active parts of the active cells, and the glial cells play a supporting role that we'll discuss in tomorrow's class. It's very important to remember that ions are impermeable to the membrane, so they won't move through. And this allows the cells then to maintain very specific concentrations of ions inside the cell. And we can then establish these ion gradients. And those turn out to be really important when we're talking about synaptic signaling. So if these are going to move through, they either have to move through transporters. Remember, a transporter everts and is only open to one side of the membrane or the other. And transporters, of course, are capable of also moving ions up a concentration gradient, so they can participate in active transport. And the other way that we can move them is via channels. Okay? And this is facilitated diffusion, but it's passive. There's no input of energy. The ions will always move down a concentration gradient. This particular example is the potassium channel, which turns out to be one of the really important channels when we're looking at an action potential. And amazingly, it's very specific for potassium. Even though sodium, Na+, same charge, similar size, sodium is mostly excluded from this channel. Okay? It's important to note that just because it's a channel doesn't mean it's unregulated. And we regulate when this thing is open and closed. And today, we'll talk about when it opens and closes, but we actually, because of the fire alarm, won't go to the mechanism until tomorrow's lecture. So here we have a diagram. And here we have all these dendrites, and these can receive signals or stimulus or whatever it might be. Within the cell body, this is just like the cell bits that we think about. So all the organelles are in here, the nucleus, the mitochondria, all that stuff's in the cell body. It then stretches out to the axon, and the axon ends at the synaptic boutons, at the synapses, and these are where the vesicles of neurotransmitter are stored, and they'll be released into the synapse to mediate effects. The axons themselves can actually be, in humans, up to around 1.5 meters, so the distance from your spine to your toes. Okay? And in mammals, at least, they're very thin, so you can't see the axonal component even though it's long. They're surrounded by Schwann cells or oligodendrocytes, and we'll talk about the importance of that later. They serve as sort of an insulating feature for this signal. So, if we want to establish a resting membrane potential, it turns out there's a charge across a membrane. And we can calculate this based on the concentration of ions inside and outside. So what we have here is squid and cat neurons. So these are common organisms for studying. And do we know why squid is common? Like, what's up with squid? Yeah. They're distributed throughout, but there's something really special about squid axons that allowed us to study them. Yeah? Yeah, they're actually, you can see them with the naked eye, actually. So you can take them out and put them in a petri dish and actually look at them, which is really cool. So they've been well studied for these, a lot of the early studies of nerves, and the ones they do today are actually very mechanical, measuring membrane potentials and things like that, and that they could be easily manipulated for that. But what's important is no matter which system we're looking at, the concentration of sodium outside is high. Potassium is high inside, so sodium's high outside, potassium's high inside, and chloride is also higher outside. Okay? And we'll see that we'll get insight into the mechanism of how cells maintain these really quite specific concentration gradients. So here is the issue. We have a membrane here, and on the tank, the cytosol, we have high potassium. And potassium carries a positive charge, so we shield that inside the cell with molecules that carry a negative charge, labeled as M minus here. And what might those molecules include? Anyone? Yeah. Yeah, so chloride's part of it. Okay? It's actually not the major one, though, if we go back to our last slide. So there's some chloride in there. What other molecules, macromolecules, maybe often have a negative charge? A negative charge. Sodium's got a positive charge. RNA, right? Lots of that in the cytosol. Actually, most proteins are negatively charged. There's DNA in the nucleus. So these are balance out the charges. So we have an issue. I'm a potassium. There's a concentration gradient. It's very favorable for me to walk out of the cell. So I leave the cell. But what I've done is I've taken a positive charge out and left a negative charge behind. So that's actually not favorable, right? So what we have is this problem because we have what's called an electrochemical gradient. The electrical from the charges and the chemical from the physical number of potassiums on each side. So what they'll do is it'll go to an equilibrium where the concentrations no longer change. And we can calculate that using this thing called the Nernst equation. And so the EX will be the potential that'll come out in volts. And we have the temperature, which will be in Kelvin. And in this case, if we're looking at mammals, it would be 37 degrees. We have the gas constant, the Faraday constant, the size of the charge, so that's going to be +1 for a potassium. And then the natural log of the concentration outside to inside. And we can simplify this. We can get rid of all this crap and just, that ends up being 0.0615 if we multiply by the log 10, rather than the natural log, of the concentration out to in volts. And so what this allows us to do then is to calculate the membrane potential if we're looking at a single ion. So I just did a couple of quick calculations to demonstrate, and these are the two that are sort of important. So this was looking at, I think this is the cat motor neuron. So if we look at the potassium, the concentration of potassium in was 140 millimolar. And the concentration of potassium out, was 5 millimolar. 5. So if you plug that in, our potential here, the membrane potential, ends up being minus 0.089, which equals minus 89 millivolts. So just remember, it's about minus 90. In contrast, if we were to do this for sodium, the concentration for sodium in was 12 millimolar. Sorry, and the concentration of sodium out was 140. And so if you calculate that, it ends up being plus 65 millivolts. So this then can be done for sodium, potassium, and chloride. But the problem is that in the cell you have sodium, potassium, and chloride. You don't have them one at a time. And this means that overall, it's much more complex. And on top of all of this, we know that the cell is working hard to maintain these particular ion gradients. Okay? And so the additional feature we have is we can see the pump here being used to maintain gradients, is that we have what are called leak channels. Okay? So a leak channel is not the same as the sodium and potassium channel that we're about to talk about. These let through a very slow stream of ions. And what's important about this is, let's say inside the cell we're trying to maintain about 140 millimolar of potassium. If too much potassium somehow gets out of the cell, it's going to be in trouble. But if we slowly have potassium leaking in, you can probably understand it actually gets easier to maintain those concentration gradients. So that makes sense, right? So there's this sort of low permeability for all three of those ions. And in this case, of course, they'll move up or down their electrochemical gradient. But how do we calculate the electrochemical gradient? This is a lot of writing that describes the Goldman equation. But really what happens is the Goldman equation takes into account that permeability, the fact that these are leaking through a little bit. And so what it does is it takes into account, if we pop right through to it, the permeability and the concentrations of all of these. Okay, so potassium, sodium chloride, but also the permeability, how much is leaking in and out at a given time. And for this, it's of course, well, not of course, for this it is a relative permeability. Okay? Meaning we assign one of these a permeability of 1, and the other ones we compare back to that permeability. Okay? So if we look at this example, if we were to say, look at this, where we've given potassium a permeability value of 1, experimentally you would need to calculate the relative permeabilities, and they find that that of sodium is 4% of the permeability, so that's 0.04, and chloride is 45%, or 0.45. So now if you plug all this in, what you find is a resting membrane potential in the cat of around, I think it's around minus 65 millivolts. And this makes sense because we know that if we're just looking at potassium and sodium, potassium was like minus 90 and sodium was plus 60. So we would expect it to be somewhere in between. Yeah?
Why is the chloride switch? Why is it in over out?
I think the chloride is switched because it's got a negative charge. And so we're calculating out by we had the charge in the first part of the equation here, but now we're actually including the charge in with this component. So I believe that's why it's flipped. Yeah. It's a reasonable hypothesis. I don't know for sure, but I think that's why it's been flipped. And so this then allows us to calculate the true resting membrane potential. Yes?
Do the resting potentials vary significantly between species?
Yeah, do they vary vastly between species? My guess is there would be some variability, but that it wouldn't, I don't think it would be that much. Like, that would be my guess, is there's not a huge variability there. Because organisms with many of them, they manage to maintain the kind of chemical levels within the cells. I think they would probably all be quite similar. Yeah. There would be definitely some variability though, but yeah. So there's a weird thing happens here. If we look at this, if something is so highly permeable that it is so much larger than all of the other. So let's say our sodium, let's say we open an actual channel. So we're basically opening a huge hole in the membrane through which massive numbers of sodium ions flood per second. That means that this permeability and this permeability basically are zero, right, at that point. And so what does that tell us? How do we describe this before? Well, basically, under those conditions, the Nernst equation works. And so what we get out of this, I think, will be obvious when we look at this slide or when we discuss this slide together. So there's two channels that are important during the action potential, the sodium and the potassium. And when these channels are open, the permeability is so high that we don't need to use the Goldman equation. We can use the Nernst equation for that particular ion, more or less. So, if we start here at a resting potential of minus 60, this is the resting potential. The way that the action potential is initiated is the sodium channel opens. And remember, when we were looking at sodium, we calculated, what did we calculate? It was plus 65 millivolts. So what happens is we start to get this depolarization occurring of the membrane. And we don't get all the way up to 0.65, because what happens is very quickly after opening, okay, this is the time in milliseconds, okay, so very soon after opening, the sodium channel slams shut. Inside the cell now, the membrane potential is positive. To get back to the resting, what happens is we open the potassium channel. So even though the concentration of potassium is higher outside, is normally, so normally I'm doing a bad job of explaining this. So what we do is we now have all this positive charge in the cell. So when we open up the potassium channel, potassium floods out of the cell. Okay? It runs down its electrochemical gradient. And we know that when we calculated it, it came out to like minus 90 millivolts. So what we call this repolarization here. So that's step 3. And we may even go to a state of hyperpolarization before the potassium channel also slams shut. Okay? So when we are down here, our potassium channel slams shut. Now, we don't have ions moving through either of these channels, and this is part of what we call the refractory period. And during the refractory period, another action potential cannot move through. What we do at this point is we kick on the exchanger, and we switch the sodium for the potassium back to those levels that we had initially. And so at this point, what happens is that we revive or restore the resting potential. And now this thing's ready to fire again. So to restore it, we have to flip our potassiums and our sodiums back. Okay? And so those are the changes then that we see during an action potential. We didn't get this far in the morning class, but I'll cover this just because we can look at it and we'll start again. Oh, did you have a question?
Sorry, I was just, what did you mean by that last part with the exchanger and flipping it?
Well, until you have it so that it's higher sodium outside than in and higher potassium in than out, it can't fire again.
Okay.
Because if you were to open up the sodium channels, again, nothing's going to come in because you've already moved your sodium into the cell. Does that make sense? And so you need to flip it and the potassium around again for it to be able to fire again. Yeah. Yes?
You mentioned that the potassium channel opens back up once the cell has a positive potential. What stops the sodium channel from opening up instead to release those positive ions?
So once this positive charge builds up, the sodium closes.
Yeah. But since they're both closed, how does the cell know to have?
The sodium needs specific signals to open. I think it's triggered by, I can't remember if it's calcium that triggers the initial signal. So it's not just a general positive charge. Whereas when we look for potassium, it's just a generalized positive charge, which we can see here. So this is looking, I don't know as much about the sodium channel. So I can't remember exactly what the trigger is. I'll find out for next class. So if we are looking at the sodium channel, this is looking at it from above. And what happens is that it's got an open channel that then is squished closed. And what squishes it closed is this helix called S4. And this helix called S4 is amphipathic, or maybe not fully amphipathic. It's got, no, it is. It's got one of the sides of it, remember they're shaped like a cylinder. One of the sides is covered with positive charge. And that positive charge, remember in the resting cell, there's all this negative charge down here, because that's the membrane potential. So that pulls that helix down and squeezes the channel closed. Once we open up the sodium channels, however, what happens is a whole bunch of positive charge has flooded in. So that pushes that helix up away from the positive charge and opens up the membrane. So that's the mechanism we have here. And so this would respond to any sort of generalized buildup of positive charge, because normally the resting potential should be negative. Yeah? Any other questions?
So in that diagram, does it flow physically through the cylinder?
Yeah, oh, sorry. So in this diagram, this kind of core section would be where the ions would potentially move through. Yeah. And so it just basically just squeezes part of the channel. We can see here, it squeezes it closed at the bottom. Yeah. Any questions? Okay, well, due to our fire alarm coffee break, I hope everyone has a good day, and we'll see everybody tomorrow.